Hi everybody, my name is Isabel Lehman, and today I'm going to be presenting our Advanced Concepts Presentation 5, and the topic is on how ATP is generated. So first I have some background information. Cellular respiration is the process that allows your body to harvest a huge amount of energy from a single glucose. In fact, it's so efficient that you get 30 ATP for every one glucose molecule you eat. So if you see in this diagram, glucose eating, cellular respiration. So in this presentation, I'm going to break down and walk you through the cellular respiration process to understand exactly where each ATP comes from. So here we have a little cellular respiration overview. The process consists of 4 steps: glycolysis, pyruvate oxidation, which is sometimes referred to as the link reaction, the citric acid cycle, also known as the Krebs or the TCA cycle, and then finally oxidative phosphorylation, which is also sometimes referred to as the electron transport chain. However, the two terms are simultaneous as they occur at the same time and are directly related to one another. So the first stage of cellular respiration is glycolysis, and it occurs in the cytoplasm. So a sequence of enzyme reactions transforms 1 molecule of glucose into 2 molecules of pyruvate. The 10-step conversion, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, requires the presence of specific enzymes: hexokinase, phosphohexosaisomerase, phosphofructokinase, aldolase, triose phosphate isomerase, glyceraldehyde 3-phosphate dehydrogenase, phosphoglycerate kinase, phosphoglycerate mutase, enolase, and pyruvate kinase. So with the help of these 10 enzymes, glucose is first transformed to glucose 6-phosphate, then to fructose 6-phosphate, then to fructose 1,6 biophosphate, glyceraldehyde 3-phosphate, and dihydroxyacetone phosphate. And then step 5, it is converted to glyceraldehyde 3-phosphate, and then to 1,3-bisphosphate glycerate, then to 3-phosphoglycerate, then to 2-phosphoglycerate, then to phosphophenylpyruvate, and then finally to pyruvate. So in glycolysis, 2 molecules of ATP are consumed in the first step and in the third step. You can see that ATP is being consumed and turned into ADP. 2 molecules of ATP are also created in glycolysis. In the 7th step, 2 ADP are created or transformed into 2 ATP, and in the 10th step, same thing. And finally, 2 molecules of NADH are also created, and that happens in the 6th step. So overall, glycolysis results in a net ATP generation of 2 ATP. This chart is basically totaling everything that we just talked about in the previous slide. So when we look at this chart, we have -1 plus -1 plus 2 plus 2, which is equal to 2. And although 2 molecules of NADH were also generated, as you can see here in this last line, you may notice that they do not count toward the total ATP, as they will be used in further steps of cellular respiration. So we will come back to this. The second stage of cellular respiration is pyruvate oxidation. And in this step, each pyruvate molecule produced during glycolysis is changed into acetyl-CoA. The reaction requires participation of 3 enzymes, which are pyruvate dehydrogenase, dihydrolypoyl transacetylase, and dihydrolypoyl dehydrogenase. And it also requires 5 coenzymes, which are thiamine pyrophosphate, lipoic acid, flavin adenine dinucleotide, nicotinamide adenine dinucleotide, and CoA. So the first step is that pyruvate loses CO2 and HETPP is formed. In the second step, hydroxyethyl group is the hydroxyethyl group, which is this HETPP that was just formed, is transferred to lipoic acid and oxidized to form acetodihydrolipamide. And then in the third step, acetyl-CoA acetyl group is transferred to CoA. And in the last step, dihydrolipamide is redoxized. Since ATP is not directly produced in this step, that is why we refer to this reaction as the link reaction of cellular respiration. And although certain sources tend to neglect pyruvate oxidation as an important stage, I felt it was necessary to include, as the process would not be able to proceed without it. So as you can see on this chart, 2 molecules of NADH are formed during the conversion of pyruvate into acetyl-CoA. However, these NADH do not count toward the total yield, as they will be used in further steps. The third stage of cellular respiration is the citric acid cycle, which begins when the acetyl-CoA made in this last step, this is the last step, combines with the 4-carbon molecule oxaloacetate, which you can see right here. They combine and go through this cycle of reactions, ultimately regenerating this original molecule. So through the process, ATP, NADH, and FADH are produced, as you can see here. NADH, FADH, and ATP. Oops, and I just blocked out this CO2, but CO2 is also released, or carbon dioxide. Because each molecule of glucose produces 2 pyruvate molecules, it takes 2 turns through this cycle to completely break down the original glucose. And the 8-step cycle requires the presence of these 8 enzymes, which are alpha-ketoglutarate dehydrogenase, succinyl-CoA synthase, succinate dehydrogenase, fumarase, malate dehydrogenase, citrate synthase, acantase, and isocitrate dehydrogenase. So, with the help of these enzymes, as I said, acetyl-CoA combines with the oxaloacetate and then gets turned into citrate and then isocitrate and then alpha-ketoglutarate, then succinyl-CoA, then succinate, then fumarate, then malate, and then back to this, and then it goes through this cycle again. The citric acid cycle results in a net ATP generation of 2, as you can see here. 2 molecules of ATP are formed from the 2 molecules of the succinyl-CoA. And although 6 molecules of NADH right here are formed and 2 molecules of FADH are formed, I could highlight those. So we have 6 molecules of NADH and this is pretty bad highlighting, but 2 molecules of FADH too. They, again, do not count toward the total ATP yield for the citric acid cycle, I should say, as they will be used in further steps of cellular respiration. The fourth and final stage of cellular respiration, oxidative phosphorylation, begins when, definitely going to need my highlighter for this, when the NADH and FADH2 made in the previous step, so here we have NADH and FADH2, deposit their electrons. So here we can see these are their electrons being deposited into the electron transport chain, and they turn these back into their empty forms. So the NAD plus, NADH and FADH get turned back into the NAD plus and FAD. So as the electrons move down the chain, energy is released and used to pump protons out of the matrix. So here we have the matrix, and then they're pumping these protons out of the matrix, forming a gradient. So these protons then flow back through the matrix through an enzyme called ATP synthase, which you can see here. So the protons come back through here to form ATP. So in order to calculate the total ATP formed during oxidative phosphorylation, we must understand the following conversion rates. NADH is typically equal to 2.5 ATP. However, for electrons that enter at the level of Q-cytochrome C oxyreductase, such as NADH transferred by the glycerol phosphate shuttle, the yield is about 1.5 molecules of ATP. And for FADH, it usually yields about 1.5 ATP. So now let's take a look at this chart. If you can see here, this first one says 2 molecules of NADH are formed in glycolysis. Each yields 1.5 molecules of ATP because yield is about 1.5 when NADH, assuming transport of NADH by the glycerol 3-phosphate shuttle. So like we said, NADH transferred by the glycerol 3-phosphate shuttle yields about 1.5. So with that, we have 2 times 1.5 is equal to 3. And then we have 2 molecules of NADH are formed in the oxidative decarboxylation of pyruvate. So I should mention that, as we noticed here, it talks about the NADH that were formed in glycolysis, and now we're talking about the NADH formed in oxidative decarboxylation. So as I mentioned previously when talking about the ATP yield of the previous steps, these NADH molecules end up coming back and are calculated in the total yield. So we have 2 times 2.5 equal to 5, and the 2 molecules of FADH2 that are formed in the citric acid cycle times 1.5 equals to 3. And then we have 6 more molecules of NADH formed in the citric acid cycle, and these are yielding 2.5, which is equal to 15. So we have 3 plus 5, which is 8, plus 3 is 11, plus 15 is 26. So oxidative phosphorylation yields about 26 molecules of ATP. Depending on whether NADH or FADH2 is employed as the electron carrier during this oxidative phosphorylation step, that is what determines whether 26 or 28 molecules are produced. However, here, 26 are produced because NADH was employed. So finally, we have the total chart, which is the ATP yield from the complete oxidation of glucose, or I guess you could say the ATP yield from the complete cellular respiration process. So we have all of the ATP generated, or I would say removed, consumed by glycolysis, then the conversion of pyruvate, so it's accounting for that NADH2, but they're not added to the total until down here. And then the citric acid cycle, the 2 molecules that were produced there, and then this is what we just covered in the last step. So when we total all of these, we have 30. And that is all I have for you guys today. So I hope you learned something new, and I look forward to hearing your feedback. Thanks.